Current developments in epidemiological forecasting have brought a renewed focus to the threat of hantavirus, as mathematical models now suggest the potential for wider transmission patterns than previously documented. While the specific data points of these new projections originate from studies investigating concentrated environments, including the unique logistics of cruise ships, the implications reach into broader public health surveillance. Hantavirus refers to a family of viruses spread mainly by rodents, and depending on the specific strain, it can lead to two major clinical presentations: Hantavirus Pulmonary Syndrome, or HPS, and hemorrhagic fever with renal syndrome, known as HFRS. These mathematical models are designed to identify how environmental variables, such as rodent population density and human proximity in enclosed spaces, might lead to spillover events where the virus jumps from its natural reservoir to humans. The mention of cruise ships in recent modelling is particularly significant for public health officials because these vessels represent a closed ecosystem where hygiene, ventilation, and vector control are paramount. Unlike many other viral threats that dominate the headlines, hantavirus is not typically known for human-to-human transmission, with the notable exception of the Andes virus strain in South America. Instead, the risk is almost entirely environmental. Public health agencies utilize these predictive models not to cause alarm, but to sharpen the focus of clinical readiness and laboratory monitoring. By understanding the mathematical probability of an outbreak, agencies can better allocate resources to high-risk zones, whether they are rural agricultural settings or, as the source suggests, complex maritime environments. The core of the concern lies in the high mortality rate associated with certain hantavirus strains, which makes early detection and the understanding of transmission pathways a critical priority for international health monitors. As we examine these new projections, it is essential to distinguish between a theoretical model's terrifying potential and the actual documented cases currently under review by global health bodies. This reporting aims to bridge that gap, providing a factual look at the science of the virus and the systems in place to prevent its spread beyond the isolated incidents often reported in the past. Understanding the mechanism of the virus is the first step in contextualizing these new warnings. The virus is shed in the urine, feces, and saliva of infected rodents, most commonly the deer mouse, white-footed mouse, rice rat, or cotton rat in the Americas. When these substances are disturbed, the virus becomes aerosolized, meaning it enters the air as microscopic particles that humans then inhale. This specific mode of transmission is why confined spaces, ranging from seasonal cabins to the utility decks of large ships, are often the focus of rigorous mathematical modelling and public health directives. The transition from mathematical theory to clinical reality involves a complex network of diagnostic laboratories and surveillance agencies. When models predict an uptick in viral activity, the first line of defense is the systematic monitoring of rodent reservoirs. Public health officials and specialized biologists conduct field surveillance to trap and test local rodent populations, seeking to determine the prevalence rate of the virus within those communities. In a maritime context, such as the cruise ship scenarios mentioned in recent reports, this involves stringent integrated pest management protocols and the inspection of cargo and food storage areas. If a model suggests an increased risk, laboratories must be prepared for a surge in diagnostic requests. Confirming a hantavirus infection is a sophisticated process that generally takes place in high-security facilities, often designated as Biosafety Level 3 labs. Clinicians look for specific markers in a patient's blood using enzyme-linked immunosorbent assays, or ELISA, to detect IgM antibodies that indicate a recent infection. Alternatively, polymerase chain reaction, or PCR, testing can be used to identify the viral RNA itself. The difficulty for hospitals and clinics lies in the early symptoms, which are often indistinguishable from the common flu, including fever, muscle aches, and fatigue. It is only as the disease progresses, sometimes rapidly, into respiratory distress or renal failure that the severity becomes apparent. This lag between initial infection and clear clinical presentation is why mathematical models are so vital. They provide a head start for doctors to consider hantavirus as a differential diagnosis in patients with relevant travel or exposure histories. Regarding the mention of trade, travel, and shipping, These sectors are under constant observation by international health regulators. The movement of goods and people provides a theoretical pathway for the movement of vectors, though most hantavirus strains are geographically tethered to the specific range of their host rodents. While the source material highlights the terrifying spread beyond cruise ships, verified data from the World Health Organization and the Centers for Disease Control and Prevention generally show that outbreaks remain localized to areas with high rodent activity. The disconnect between terrifying predictions and historical data often stems from the variables used in modeling. Researchers may look at worst-case scenarios to ensure that public health infrastructure is never caught off guard. Currently, there are no reports of a widespread, multinational outbreak linked to maritime travel, but the vigilance within labs and transit hubs remains high. Officials emphasize that the accuracy of any mathematical model depends on the quality of the data fed into it, which is why consistent reporting from rural clinics and port authorities is a cornerstone of modern infectious disease management. Prevention and public guidance remain the most effective tools in the management of hantavirus, regardless of what mathematical models might project for the future. Public health agencies consistently advise that the primary way to avoid infection is to eliminate or minimize contact with rodents in and around human environments. For travelers and those living in areas where hantavirus is endemic, this involves sealing up entry holes in buildings, using traps to manage rodent populations, and keeping food sources in tightly sealed containers. A critical aspect of hantavirus prevention that is often overlooked is the proper way to clean up infested areas. Because the virus is transmitted through aerosols, sweeping or vacuuming dry droppings can actually increase the risk by kicking viral particles into the air. Instead, official guidance suggests saturating the area with a disinfectant, such as a mixture of bleach and water, for several minutes before wiping it up with paper towels. This focus on low-tech prevention stands in contrast to the high-tech nature of the mathematical models, but it is the most reliable method for reducing individual risk. Regarding the limits of our current knowledge, it is important to state that while models can predict potential spread, they cannot account for every environmental variable or human behavior. There is a degree of uncertainty inherent in any epidemiological forecast. A predicted terrifying spread may never materialize if rodent populations crash due to weather changes or if public health interventions are successful. Furthermore, there is currently no specific vaccine or cure for hantavirus infection in humans. Treatment is primarily supportive, often requiring intensive care and mechanical ventilation to manage the respiratory symptoms. This lack of a specific pharmaceutical intervention is why surveillance and prevention are the pillars of the global response. Honest reporting acknowledges that while the virus is a serious health threat, it is also a rare one. The global health community continues to monitor for any changes in the virus's behavior, such as mutations that might allow for easier human-to-human transmission. But such changes have not been documented in the strains currently causing concern in the Northern Hemisphere. In closing, while mathematical models serve as an essential warning system for agencies and maritime operators, they are tools for preparation rather than a guarantee of a coming crisis. Public health officials urge a calm but informed approach. Stay aware of your environment, follow local health directives regarding rodent control, and seek medical attention if symptoms appear following potential exposure. You can stay informed on further public health updates by subscribing to this channel. The transition from theoretical mathematical modeling to tangible clinical surveillance requires a highly coordinated effort between international health agencies, port authorities, and diagnostic laboratories. When a model indicates a potential for expanded transmission, as suggested by recent data concerning maritime environments and cruise ships, the first response involves a rigorous assessment of current vector control protocols. Public health officials emphasize that while a model might forecast a terrifying spread, the actual containment of hantavirus depends heavily on the integrated pest management systems utilized by the shipping and travel industries. These systems are not merely about sanitation. They involve the structural auditing of vessels to ensure that rodent reservoirs, primarily various species of mice and rats, cannot establish nesting sites in cargo holds, ventilation ducting, or food storage areas. If a cruise ship or a commercial vessel is identified as a point of interest, international health regulations dictate a specific sequence of inspections and possible quarantines. This intersection of trade and public health is a critical development because the movement of goods provides a theoretical, though historically rare, pathway for the relocation of infected rodents or contaminated materials across geographical borders in the laboratory setting. The development of hantavirus diagnostics has become a cornerstone of modern infectious disease surveillance. Because the symptoms of Hantavirus Pulmonary Syndrome and hemorrhagic fever with Raynaud syndrome often mimic common respiratory or gastrointestinal illnesses in their early stages, clinicians must rely on sophisticated molecular testing to confirm a diagnosis. This process typically occurs in Biosafety Level 3, or BSL-3, facilities, which are equipped to handle airborne pathogens safely. Laboratory technicians utilize enzyme-linked immunosorbent assays, known as ELISA, to identify the presence of IgM and IgG antibodies, which signal that a patient's immune system is responding to a recent or past infection. Additionally, the use of polymerase chain reaction, or PCR, testing allows for the direct detection of viral RNA in blood or tissue samples, providing a definitive confirmation even before an antibody response is fully developed. The logistical challenge for hospitals, particularly those in port cities or rural regions, is the speed of these tests. While rapid diagnostic kits are in development, most confirmed cases still require the transport of samples to centralized state or national reference labs, creating a period of clinical uncertainty where medical teams must treat the patient based on symptoms alone. Furthermore, the core of recent developments lies in the contrast between these high-end mathematical projections and the verified epidemiological data maintained by organizations like the World Health Organization. While the source mentions a spread beyond cruise ships, health agencies generally maintain that hantavirus remains a disease of environmental focal points. This means that transmission is usually tied to a specific, localized environment where rodent density and human activity overlap, rather than a broad, person-to-person pandemic scenario. Agencies monitor these focal points through sentinel surveillance, where rodent populations in high-risk areas are regularly trapped and tested for the virus to determine the prevalence rate. If the percentage of infected rodents in a specific area rises significantly, health departments issue localized warnings. The mention of cruise ships in recent modeling highlights a specific concern regarding closed ecosystems, where recycled air and shared living quarters could theoretically amplify the risk of aerosolization if a vector is present. However, hospital data from global maritime hubs has not yet shown a statistically significant increase in cases linked to shipboard travel, suggesting that current mitigation strategies, such as high-efficiency particulate air filtration and strict waste management, are functioning as intended. The diagnostic and surveillance infrastructure is also tasked with investigating the spillover mechanism, the exact moment the virus jumps from its natural host to a human. This involves not only biology but environmental science, as changes in climate or local ecology can drive rodent populations into closer proximity with human structures. When agencies analyse the terrifying potential of a spread, they are often looking at these ecological triggers. In a laboratory context, this means sequencing the viral genome to ensure that the virus is not mutating in ways that would increase its stability in the environment or alter its path of infection. The clinical reality for patients remains serious. Once the virus reaches the lungs or kidneys, it can cause rapid capillary leak syndrome, leading to the severe symptoms that give the virus its high mortality rate. Therefore, the development of better bedside monitoring and the integration of model data into hospital triage systems are currently the primary focuses of the medical community. By treating the mathematical model as a weather forecast for viral risk, public health officials hope to narrow the gap between the emergence of a threat and the implementation of a clinical response, ensuring that shipping lanes and travel corridors remain monitored and secure. The transition from predictive modeling to practical public health action relies on the clear communication of prevention strategies and a grounded understanding of the virus's environmental constraints. While mathematical models may project a wider distribution of hantavirus, the primary defense for the general public remains the rigorous management of the rodent-human interface. Public health guidance is uniform across international agencies. The most effective way to prevent infection is to eliminate rodent access to human living and working spaces. This involves a multifaceted approach known as rodent proofing, which includes sealing any gaps or holes larger than 1/4 of an inch in building foundations and walls using metal flashing or heavy-duty mesh. Furthermore, the management of food sources is critical. All pet food, birdseed and human provisions must be stored in thick plastic or metal containers with tight-fitting lids. In the context of the maritime and shipping industries highlighted in recent reports, these protocols are scaled up to include the inspection of shipping containers and the use of specialized rat guards on mooring lines to prevent the migration of rodents from ports onto vessels. A critical takeaway for the public involves the specific mechanics of cleaning areas where rodents have been present. Because hantavirus is transmitted primarily through the inhalation of aerosolized particles from rodent urine, droppings, or nesting materials, the act of cleaning can itself become a risk factor if performed incorrectly. Public health officials strictly advise against sweeping or vacuuming dry rodent waste, as these actions launch viral particles into the air. Instead, the standard protocol is wet cleaning. This requires the use of a disinfectant, ideally a solution of 10% bleach and 90% water, which must be sprayed directly onto the contaminated area and left to soak for at least 5 minutes. This process inactivates the virus before it can be physically disturbed. Only after the material is thoroughly saturated should it be wiped up with paper towels or a cloth, which are then disposed of in sealed bags. For those entering confined spaces that have been closed for long periods, such as seasonal cabins, storage sheds, or utility compartments on ships, the guidance is to ventilate the area for at least 30 minutes by opening all doors and windows before entry, and to utilize personal protective equipment, including gloves and N95 respirators. If the level of infestation is significant, honest public health reporting also requires an acknowledgement of the limits of current medical knowledge and the uncertainties inherent in epidemiological modeling. While we have identified the various strains of hantavirus and their respective host rodents, there is currently no commercially available vaccine for hantavirus in the United States or Europe. Nor is there a specific antiviral treatment that can cure the infection once it has reached the critical stage. Medical intervention is almost entirely supportive, focusing on oxygen therapy, fluid management, and in severe cases, the use of extracorporeal membrane oxygenation, or ECMO, to bypass the lungs and heart. This lack of a pharmaceutical silver bullet is exactly why mathematical models that predict a spread are treated with such gravity by health officials. Without a vaccine, prevention and early detection are the only viable tools for reducing mortality. However, it is equally important to contextualize the terrifying nature of these models. Most hantavirus strains are not easily transmissible between humans, meaning that even if the virus moves into new areas via shipping or transit, it does not typically lead to the kind of rapid person-to-person outbreaks seen with respiratory viruses like influenza. The risk remains tied to individual environmental exposure. As we